Hello everyone, welcome to Dr. Osman Setu Trends, Swing into Educational Trends. Today we will be talking about prodrug in medicinal chemistry. The term prodrug was coined by Adrian Albert in 1958, and prodrugs are actually defined as the conversion of biologically inactive compound to active compound by metabolic biotransformation at a specific site and time. Examples, we have castor oil, which exhibits laxative nature after it is hydrolyzed in the intestine to the active racilonic acid. Another example is the conversion of prontosil, a drug, to sulfanilamide. The designing of product is essential in order to eliminate unwanted pharmacokinetic and pharmacodynamic drawbacks. So, as I told you before also, pharmacokinetic and pharmacodynamic drawbacks, that is what drug does to the body and what body does to the drug. These includes the unpleasant taste, odor, gastrointestinal effects, limited duration of action. There could be instability, there could be non-specificity, there could be poor absorption, poor distribution or poor water solubility, or sometimes inability to diffuse. If we have an idea about ideal properties of product, we already know that any product should be pharmacologically inert. It should not have any pharmacological action. A product is inactive or less active than the parent drug. The linkage between the drug and the carrier, a carrier is the one which carries the drug to that particular site of action. So, the linkage between the drug and the carrier must always be easily cleaved in vivo, inside the environment, like at the site of action it should be easily cleaved, there should be no difficulty there. The carrier molecule must be non-toxic, like it should not have any toxic effects, because its job is to carry the drug, that's it. It should not produce any toxic pharmacological actions there. Chemical or enzymatic transformations into active form should be rapid at the target site. They should be rapid because duration of action should be initiated, and onset of action should be according to the required pharmacological action. Apart from drug, the metabolic fragments of carrier molecule, because carrier molecules will be forming some metabolic fragments, so that metabolic fragments should be non-toxic and are to be rapidly eliminated. They should not be there in the body, right? They should be rapidly eliminated because they may create some pharmacological actions, then again that will be toxicity production. Now, if we talk about the classification of prodrugs, prodrugs can be classified based on structural modifications and based on site of conversion. If we talk about structural modifications, then we have prodrugs of two types, that are carrier link prodrugs and second type is bioprecursors. Carrier link prodrugs can also be divided into three, that is bipartite, the first category, the second is tripartite, and the third is mutual. Now, if we go to the prodrugs classification based on site of conversion, we will have type 1A and type 1B. Now, let us talk in detail about it. The first type of classification based on structural modification, we have the first type, that is carrier link prodrugs. Carrier link prodrugs consist of the attachment of a carrier group to the active drug to alter its physicochemical properties. The subsequent enzymatic or non-enzymatic mechanism releases the active drug moiety. Now, you can see here the active drug and inert carrier are made as prodrug with the covalent bond. So, the covalent bond is broken by the chemicals or the enzymes which are present in the environment at the site of action, and you will get the active drug and the inert carrier at the site of action. Hydrolysis reactions must take place at an adequate rate. We have examples of drugs called becampicillin or progabalin. Now, if you talk about the first category or the first sub-prodrug in carrier linked prodrugs, that is bipartite prodrug. Bipartite product is composed of one carrier or group attached to the drug. Such products will have greatly modified lipophilicity due to the attached carrier. The active drug is released by hydrolytic cleavage either chemically or enzymatically. Example here we have is tolmetine glycine prodrug, which can also be further subdivided into tolmetine and glycine. We have second type of carrier link product, that is tripartite product. Here the tripartite product is the drug linking structure carrier. The carrier group here is attached via a linker to a drug. There will be always a linker. So that linker is in structure. Moving on to the third type of carrier link product, that is mutual product. So mutual product consists of two pharmacologically active agents coupled together so that each act as an promoiety for the other agent and vice versa. A mutual product is a bipartite or tripartite product in which the carrier is synergistic drug with the drug to which it is linked. You can see in the figure how the parent drug is reacting on biological action and how it is excreted and how the mutual product is arranged. Now, an example for this we have is benorilate, which is a mutual product of aspirin and paracetamol. We also have another example, that is sultamycin, which on hydrolysis by an esterase produces ampicillin and sulbactam. Now moving on to the second type of product under structural modifications are bioprecursors. These are devoid of a barrier linkage and they produces their effects after in vivo chemical modifications of their inactive form. Bioprecursors product rely on oxidative or reductive activation reactions, unlike the hydrolytic activation of carrier link products. They metabolized into a new compound that may itself be active or further metabolized to an active metabolite. Example we have is prontosil, which is inactive and it gives sulfanilamide, an antibacterial drug, as well as another drug. Also we have cyclophosphamide, which is inactive and gives phosphoramide mustard, that is active compound, and the enzyme here is a liver enzyme. Now the second main reason, based on the site of conversion, type 1 prodrugs are metabolized intracellularly. In that we have type 1A product which are metabolized at the cellular targets of their therapeutic actions. Example we have acyclovir, cyclophosphamide, L-dopa and zidovudine. We also have type 1B product into this category, which converts into parent drug by metabolic tissues, namely by liver. Example carbamazepine, captopril, heroin, primidone. We have another category, the type 2-1, which are metabolized extracellularly. In that we have type 2A, which are metabolized in and around gastrointestinal fluid. Example we have loperamide oxide and sulfasalazine. We have type 2B within the circulatory system or other extracellular fluid compartments. We have type 2B products which are metabolized within the circulatory system and or other extracellular fluid compartments. Example aspirin, phosphonitoin. We have type 2C also, which are metabolized near the therapeutic targets or cells. Example we have antibody directed enzyme prodrugs or gene directed enzyme prodrugs. Now moving on to carrier linkages of various functional groups. If we talk about carrier linkages of various functional groups by taking examples like carboxylic acids and alcohol, these can be prepared by conversion to an ester. The ester can be easily hydrolyzed by esterase enzymes present in plasma and other tissues to give active drug. We have carrier linkages of enzymes like esterase, hydrolase, lipase, cholesterol, esterase, acetylcholinesterase, carboxypeptidase. We can see here example of chloramphenicol palmitate. With the help of esterase, it gets converted to chloramphenicol and palmitic acid. Another example we have is enalapril, the most widely prescribed ACE inhibitor. It is the ethyl ester product of the active diacid enalaprillate. We have the third functional group, that is amine. Amines are not derivatized to amide prodrugs due to high chemical stability of the amide linkage and lack of amidase enzyme. Hence, manic bases can be utilized as prodrug forms of amines. You can see here ampicillin reacted with acetone gets converted to hetacillin. Now we have the fourth functional group, that is azo linkage. Amines are derivatized to azo linkage prodrugs. Example prontosil. You can see prontosil, which is inactive. With the help of azo reductase, it gets converted to sulfanilamide antibacterial. and other intermediate. I have given this example before also. The fifth functional group we have is carbonyl compound. Aldehyde and ketones are converted to prodrug. These have generally involved derivatives in which sp2 hybridized carbonyl carbon is converted to an sp3 hybridized carbon attached to two heteroatoms such as oxygen, nitrogen or sulfur. Example we have is methanamine, which releases formaldehyde in urine, which act as an antibacterial agent. Now let us talk about the applications of prodrug in drug designing. How the prodrug concept is important in drug designing. Prodrug concept can be used in the alteration of solubility. Example we have is acylation with aliphatic or aromatic carboxylic acid groups, which decreases the water solubility and enhances lipid solubility. Now we can see that with the help of prodrug we can alter solubility. So if we want to increase the lipid solubility of the drug, so we can improve the lipid solubility by decreasing the water solubility. Example here is acylation with aliphatic or aromatic carboxylic acids. If we want to increase hydrophobicity of the drugs, then we can go with the formation of hemisuccinates, hemiglutamates, hemithalates, metasulfobenzoates, etc. These particular groups of structures serves as the site of formation of water soluble sodium, potassium and amine salts. Now if you want to improve absorption and distribution, then the third application of prodrug is, example here is an esterification and acidonidation, which improves the topical absorption of corticosteroids, which serves as an anti-inflammatory drug and also used in allergic and pruritic skin conditions. Example of the drug is fluocinolone acetonide and fluocinonide. The oral absorption of ampicillin is more in its ester form, such as pivampicillin and becampicillin. Now if you want to prepare site specific products, then centrally acting drugs must be designed in such a way that they easily cross blood-brain barrier. This can be made possible by attaching a lipophilic carrier to the hydrophilic drug in a closely bond form. Example, beta-lactam antibiotics. A hydrophilic drug is used in the treatment of bacterial meningitis, which can cross blood-brain barrier. Because of its hydrophilicity, its entry into the brain is slow. Prodrug dihydropyridine penicillin delivers beta-lactam antibiotic in greater concentration into the brain. The neoplastic cells possess number of phosphamidases compared to normal cells. Thus, the cyclophosphamide serves the purpose of anti-neoplastic agent. The expected action of the nitrogen mustard is hydrolyzation in tumor cells. Progabite is a prodrug of gamma-aminobutyric acid, an inhibitory neurotransmitter. Oxyphenicetine, a sterilant of bowel, is an active form if it is administered via the rectal route. The orally active prodrug is designed in order to release the active oxyphenicetine through hydrolysis. We can design a prodrug which is active at the specific site of action. Example, diethylstilbestrol diphosphate. It is a prodrug of diethylstilbestrol. It has been designed for its site specific delivery to prostate carcinoma tissues. This is based on the notion that phosphates and amidases are present in greater amounts in the tumor cells. Now, design of stable prodrugs. The efficacy of certain oral drugs decreases because of the first pass effect. We all are aware of that. The metabolic studies help in designing prodrug for better absorptions of such drugs. Example is the prodrug of propranolol, which is propranolol hemisuccinate, which gets metabolized to propranolol O-glucuronide, parahydroxypropranolol and parahydroxypropranolol glucuronide. Thereby, an oral administration propranolol hemisuccinate enhances the plasma levels of the parent drug propranolol nearly 8 times. Now we have another application of prodrug is this design of slow and prolonged release drugs. We can say novel drug delivery systems. For prolonged release, the aliphatic ester drugs are suitable since they hydrolyze slowly. This concept is applied in the design of sex hormones and their derivatives. Example we have is haloperidol. If it is administered alone, exhibits a duration of action of just over 2 to 6 hours. However, when haloperidol decanoate is administered by the solution of intramuscular route in sesame oil, the activity of the drug remains about for a month. So, from 2 to 6 hours to 30 days, that much duration can be adjusted. Now we have design of drugs with lower toxicity. If you want to get drugs with the lower toxicity, then esterified aspirin produces less gastric effect when compared to aspirin, that is gastric irritation bleeding. You all are aware that aspirin can give a side effect of gastric irritation. So esterified aspirin can be produced. Now we have another application of prodrug is products for better patient compliance. Clindamycin is an example. Clindamycin, a drug with bitter taste, is converted to clindamycin palmitate ester by increasing the chain length of two acyl esters of parent drug. The bitter taste of clindamycin is due to the water soluble product form when the drug is dissolved in saliva. So this solubility gets decreased by esterification, hence the drug is rendered palatable. Now let us talk about the advantages and disadvantages of prodrugs. Advantages of prodrug concept in drug designing is like the patient acceptability. It can be improved. The administration and formulation of the drug can be improved. The drug can be more permeable and more absorbed. Then the fourth one is you can change the distribution profile of the drug. The fifth advantage is the drug protection from rapid metabolism, and the change in elimination can also be done with the help of prodrug concept. The seventh advantage of prodrug concept is we can overcome toxicity issues, another added advantage. Then you can design prodrug which are site specifics, and you can improve the drug stability and solubility of prodrugs. If we talk about the demerits or disadvantages of prodrugs, then with the usage of prodrugs, there may arise a chance of generation of toxic metabolites which are not generated when the parent drug is used. The second major disadvantage of prodrug is the metabolites produced by prodrugs and carriers moiety may alter the pharmacokinetic features of parent drug by enzyme inhibition or enzyme induction. The third major disadvantage of prodrug is the toxic metabolites may also get generated in case the carrier moiety could not remain inert in nature. So this completes prodrug in drug designing. Thank you.